interference. So stem cells, before we talk about stem cells in this lecture, basically we are going to talk about two things in major rather than only stem cells. The first thing that we are going to talk about is potencies. So what is meant by potency, or the plural is potency? Potency is actually the fate or determination or differentiation among cells. And potency, it refers to the possible available number of fats. This is actually wrong. This is, let me have a look. This is F-A-T-E, fates a cell can acquire. Generally, it is categorized by the potential, like a cell has a potential, but using this potential, it can be changed into, differentiate into other types of cells. And sorry. So it's a very important concept to develop, because in general, it is known very clearly that potency would decrease with increase in age, and it will also decrease as different parts of the body specialize. It means when a body has developed a special organ or special type of cells, they perform their particular function, then the potency will decrease a great deal. This is true for all human eukaryotic cells or higher eukaryotic cells, but only one cells, and these cells are known as stem cells. We still haven't yet defined what are stem cells, but what is meant by this notion is that potency for all eukaryotic cells and organs would diminish, or would be decreasing with the age. As the age increases, the potency decreases. As different parts of the bodies specialize to become dedicated cells or organs, their potency would decrease. But these two cases would not be applicable in case of stem cells. Irrespective of their age, they will still be very potent. Classification of potencies, actually the potencies can be divided into 4 different types: totipotency, pluripotency, multipotency, and unipotency. And the cells are consequently called as unipotent or totipotent or pluripotent. Totipotent cells we have already discussed in detail in case of plant biotechnology, where we discussed it in the tissue culture part. Ability of any vegetative cell or any cell of the organism to differentiate into a whole organism. When we talk about eukaryotes and animals, so we say it's an ability to differentiate into all possible cell fates. For example, a zygote. What is a zygote? The zygote, in its very early stages of development, it's a cell, right? It's just a cell, and it has the capacity to be actually develop into a whole embryo. Pluripotent is the ability to, like, it can acquire, the cells that are pluripotent, they can acquire many fates, but not all. So it can differentiate into almost all cell types, but not like totipotent. They do not have each and every way open to them like the totipotent. And when the cells could have the ability to differentiate into a closely related family, for example, the hematopoietic stem cells, we call them multipotent. When there's their progenitor cells. Unipotent is, of course, when you have only one fate, one door open, the ability to produce cells only of your own type. But there is one difference, which, like, all cells can divide and produce cells of their own, but then when it comes to stem cells, they would have the property of self-renewal. Okay, this is an image where you could see a totipotent cell, and the totipotent cell is a zygote here. The zygote, when divides, it can divide into embryonic, pluripotent embryonic stem cells. the endoderm line, the mesoderm line, and the ectoderm line. Now, these cells, they are totally pluripotent. They have many fates open to them, but this one has all fates open to them. Then this ectoderm cell line, ectoderm line, it will divide into different cells, similar to the mesoderm, similar to ectoderm. And now these are the multipotent cells, but now here we have either bipotent or monopotent, also known as unipotent cells. Okay, carrying on. Understanding potency. Now, zygote is a perfect example of totipotency. But again, even in the zygotic cells, in the zygote cells of different animals, there would be differences. And because when we say differences, we're not talking about the physiological or the anatomical differences of zygotes. Of course, they are always there. We talk about the differences in terms of potency, or we could say in terms of totipotency. For example, two very common examples are those of armadillo and mice. In case of armadillo, this is very common, which happens in armadillo almost every single time inside the uterus of a female armadillo. When the embryo reaches the 8-cell stage, it will break into 8 different pieces, and each different piece will develop into a new armadillo and a new young offspring. Now, what does it mean? It means that even at the 8-cell stage, the zygote at 8-cell stage still is totipotent, because this is the reason why is breakage of one embryo, 8-cell stage embryo into 8 pieces will give rise to 8 new offsprings. The case in mice, mouse or mice zygote is totally different. A zygote cell in mouse at 1-cell stage is totipotent. The moment it becomes a 2-cell stage, it will no longer be totipotent. It will no longer hold the potential to be totipotent. This is the reason why we always never have 2 identical mouse or mice twins, because normally when the cell breaks, a few cell stages, a few cell divisions have already occurred, like in this case, 8-cell stage. Imagine if an armadillo cell at embryo at 8-cell stage break into 4 pieces, then there will be 4 armadillo. If it breaks into 8 pieces, it will be 8 armadillo. More than 8, it cannot, because there are only 8 cells. But less than 8, it will always be the number of, like, if this breaks into 6 cells, it will be 6 armadillo. But the problem here in the mice, the moment it gets to the developmental stage, when it's a 2-cell embryo, it is no longer totipotent. It might be pluripotent, it might be multipotent, but it's not totipotent anymore. Okay, so now let's define stem cells. What are stem cells? The classical and the, how can I say? The simple example of stem cells is they are cells with variable potencies, as we discussed. A stem cell could be totipotent, could be pluripotent, it could be unipotent, it could be bipotent. They are cells with variable potency that can self-renew. Normally found in all parts of your body. Do not think of them as some alien cells or some special out of the world type of cell. They are normally found in your body. It's just a part of us. So each and everyone in our body have stem cells. What is exciting about them is that we can harness them, we can exploit them for therapeutic purposes. In this perspective, we call them therapeutic stem cells. This is true or false? Can we actually use these stem cells in therapy? Well, the answer is yes, and the answer is also no. Because in some cases, these therapeutic stem cells really work, while in some cases it's very difficult. So I do not say it does not work, but it is so difficult to put them work that we can neglect the weaker proportion of putting them to work. So in some cases they work, and in some cases that do not work. That's why I say yes, it doesn't, no, it doesn't. In case of hematopoietic stem cells, the bone marrow transplant is a classical example that it works. We all know about it. And the other example is the skin graft stem cells. Like in this image, you can see you can harvest skin from the unaffected area of a burn victim. For example, somebody has a very severe kind of burns, and in this case, the skin actually heals really, really slow. So you want, normally when you have a wound, it starts healing from the sides. And if it's a big wound, it will really take a huge amount of, take a long amount of time. So the slower the wound heals, the more the chances of getting infections inside the wound. That's why we use stem cells. So there are three methods of this. Harvest the skin cell from the burn patient from an area where there is no burn. We expand them, we culture them in petri dishes or in culture vials using a solid scaffold. Without solid scaffold, these cells won't grow. We always discuss it. We had already discussed this. These are anchors dependent primary cell lines, so they need solid support, as we discussed in the cell cultures lectures. Normally a wire mesh, if you can see in this one, a wire mesh is used, a gauze, we can call it a gauze. And once you have a number of grafts already prepared in the lab, you can just graft, you can place them on the burn patients, and this will cause the skin injury to repair faster and avoid a number of infection chances.